Yeah, so Dave Jett, and I'm working at the National Institute of Health and Neurological Disorders. Yeah. Okay. Yes, neurological disorders or stroke. I'm the third of the three places that NIH has brought to here, and you know, it's basically our mission is fundamental knowledge, a lot of research in that area, but also overall we're just looking to reduce the burden of neurological diseases for all people. So the one other thing I wanted to mention is, you know, this will be a little bit different from what you've been looking at today, and yesterday for that matter. Two things I'm going to, well, first let me just say that I am not an expert on AD and Alzheimer's disease. I am basically working with everybody within our, you know, Parkinson's disease, ALS, everything, working with everybody. We've got a fantastic group who are experts on AD. And I actually work with them and lots of others. And let's see, the other thing I was going to say is we do have, as you know, NIA and NIEHS, and they are surely the leaders of both the for Alzheimer's disease as well as the environment. And what we're here to do is help them and do our bit in this area. So let's see, other thing. So what I'll do is just give you a few words on what we're doing in AD-ADRD to just focus on AD-ADRD and doing some specific things on what we're funding actually. So let's go ahead. First, let me just talk a little bit about the office before we start that part. So this is a paper we did a while back to try to talk about what we think. We put this in the journal Neuro journal, and what we think this is, and what we're talking about, which is definitely non-inherited factors, and what we put it in three different areas. One is the endogenous factors, such as, of course, we put in genes as well there, metabolism, microbiome, biotics, and then exogenous things, of course, like environmental toxicants, but things also like light and noise. Absolutely, climate change. It's just the biggest thing that I think about more than anything right now. And then we separated the behavioral factors into a specific thing, and of course, psychosocial effects, and also the things like lifestyle, sleep, very, very big, and also stress. And we'll have a little bit on stress that I'll talk about. So that's that. Another thing is, so what we do at a lot of the ICs at NIH is you do these strategic planning things, and for the office my office is really only about 3 years, 2 to 3 years, and so we needed to do a strategic plan on it, and it was actually postponed for a while because we had so many other strategic plans going on, and you put together this working group of council to identify research priorities. What's going to be your priorities? And we have 3 people that are in this room that are on that, and that's Amy, Gary, and Bob, who are just doing a remarkable job helping us figure out what we want to really do, and others as well. And some of the goals that we have right now, they may be shifted around a little bit, but right now is just better define what part of is within our mission. Obviously, it's going to be related to the brain and the nervous system. Identify, prioritize areas of neural exposome research, identify the best practices for communicating with and engaging neuroscientists. This is huge because, as we said before, they're just, I mean, I can't tell you how many neuroscientists I've talked to, and the first thing they say is exosome. They say, no, it's not that. But yeah, so that's a big one. Let's see. Collaboration between our NINDS as well as some of the other NIH institutes is absolutely a major thing that's going to make this work. And then tools and resources. Okay. So what have we been doing in that part? Well, I have really pushed trying to get more neuroscientists involved. We've had actually 3 things that we've done within Society for Neuroscience events. One of them is we're getting ready to do on the microbiome and ADHD in 2024 one. We did other things like, this sort of neural exposome, what is it, kind of thing. We did some other things as well. We did some work at Society of Toxicology, same sort of thing, to sort of talk about what we're doing. We've had workshops, one on team science, as you all know. We gotta have team science or there's no chance of doing this. And then another one we did on the tools and resources that we need to do it. Then other things, we did an RFI, and some other kinds of things that we've done. Webinars, we've done a request for information, and so forth. Let's see. We've been lucky enough to work with some of the ICs, especially NIEHS, on some of the things like climate change and working with the ECHO group. And I'm going to talk about some of these other things. One thing that I probably won't talk about is we've been working with biopsychosocial factors and social connectedness, and we've actually funded 1 or 2 of these, working with an office at the NIH. So forth, and other things like this. We did something with pain research that we are in charge of, and other things. The Center of Exposome Research. So we've been really, really lucky to work with some of the ICs, and I hope we can continue doing that. Okay. All right, so let's go in and we'll go. I'm going to go real fast. And just talking to you, these are the kind of things that we're funding right now. And the thing is, if you want to know more about them and it doesn't make sense what I tell you, please look at the NIH reporter and you'll have it'll tell you exactly what they're doing. So the first one we did was impact of the microbiome, gut-brain axis in AD/ADRD, support mechanistic research, mostly focused on a more rigorous and in-depth examination of the potential interactions between the microbiome and genetic and non-genetic targets. And what's wrong with that? And what's wrong with it is not really exposomes. Now, I'll tell you this, I'm not proud of it, but for most of, not all of these, I'll say, okay, you need to work on the microbiome, but you also need to add something in there as well, and not just the microbiome. And so I feel real good about that. That's going to do it. At the last minute, I'll say, well, you can just do one if you don't have anything else to go. And people will say, it's easier, right? It's easier, so they'll go for that. Now, we've had some people that know what we're talking about, and they will put other things that are not just social factors and microbiome, things like that. And that's getting closer and closer to what we think we can do when we're talking about. Okay, so let's just look at some of them. I'm not going to go through all of these, but here's one, microbiome gut dysfunction in prodromal and symptomatic Lewy body disease. And basically, this is a sort of a complex one, but, and these are probably only in their second year maybe at this point, but hopefully they'll see this SBR dysbiosis associated with increased bacterial translocation, which will have an effect on LBD. I'm going to go quick because I got a lot of them, probably too many of them. But, so another one is on the contribution of the oral microbiome. Do you remember we've got an oral microbiome, and hopefully they will look neuroinflammation pathway largely drives the relationship between periodontal disease and Alzheimer's disease, and see what that's going to look like. Okay, circadian rhythms to optimize the gut-to-brain signaling for AD, and they're looking at the role of the microbiome and circadian rhythm disruption in affecting the onset or progression of AD. Hopefully, a healthy gut microbiome and circadian rhythms will delay the onset. Another microbiome. Bottom line is they're looking for signaling under the microbiome gut axis for Alzheimer's disease. And so if you look at the AIM-3, it will conduct supervised analysis of gut microbiome metabolite biomarker discovery for prediction and modeling of clinically important pathological features. So this is sort of more like trying to see what we can actually do for this in terms of helping this. All right, so there's another one, there's only three I think, hopefully. This is rhinotoxicants, I think was one of the first ones, looking at mechanistic and early translational research focused on a more rigorous and in-depth examination of the potential interactions and how it works with genetic and non-genetic molecular targets. And here is one on particulate matter and its interaction with APOE in neurodegeneration. Bottom line is they are trying to, their objective, I guess, is to determine the mechanism-based toxicity of this particulate matter. So, another one, this one is on ultrafine particles, and they want to see, if it's lodged in the brain, cause cognitive decline leading to AD. And see, the AIM-3 initiate exploratory studies of humans, brain samples to determine whether UFPM in select brain regions of LDC in the individual are related to pathology and other things as well. But again, remember, a little more of this go to the part that I told you. Okay, more. This is on neurotoxicity due to environmental complex metal mixtures. The hypothesis is metal mixtures relative to single metals or belong to regulatory limits, impairs brain development and accelerates cognitive decline, degeneration, and eventually AD. And, you know, looking at gene-environmental interactions is what they're trying to do in A3, which I'm sure they'll do. Air pollution, tabulum, and Alzheimer's disease in Mexican Americans. And this is, you know, looking at metabolics approach to find biomarkers for air pollution toxicity. This may be the last one for that. Synergistic white matter injury from diesel exhaust. So this is diesel exhaust. And let's see, hypothesis is diesel exhaust will increase important genes that will directly interact with the others. Oh, a lot. This one is on association of utero, exposome with life course cognition and prodromal AD. At last name determine if adolescent cognition associated with prenatal exposome. Did a lot for this one. And mechanisms of gene-environment interactions development in lead exposure. We all know that that's a big issue. So, and again, let's just go to the NIH report if you want to read more about those. So we did have a one on the role of environmental stress in the health inequities of ADRD, and it supported mechanistic, translational, and human subject studies to understand the relationship between environmental stressors and the health inequities. And I think I took them out because they're not on the recorder yet, so I couldn't. Yeah, I didn't put those on yet. Recorder, I said, that's a recorder. So that's one thing we did. We got a lot of people interested in that. A lot of, hopefully we'll be able, I think we're going to be able to fund about 5 or 6 of those. Now we have 2 other ones that are coming out now. Hopefully we can get back and get this going. One of them is on interaction between environmental factors and Lewy body dementia, and it's, you know, it's simple that we're just going to use to look at modifiable targets for Lewy body dementia, and hopefully it will support research that will firmly establish these causal links. Causal I use with at least close to causal links between environmental factors and Lewy body. And then the other one that's coming out with it is risk and disease following nervous system exposures at biological interfaces. Now, we've sort of done this before, but sometimes some of these things that are out there, they never even had any. It doesn't work because it's not on some of these biological interfaces. It's just on other things, and the nervous system never even sees it. So we wanted something that was going to be specific about that. Determining how exposure can vary in the surfaces. Includes toxins, toxic chemicals, viral and other pathogens, so it's sort of broad. And that's it in terms of what I have for you. And I'll try to answer if you have any questions. This is kind of different